Thanks for the invitation, first of all. And I'm happy to be here to talk a little bit about brain stimulation in general. So today there are two sessions. So this is the first session, and I'm going to first of all introduce different brain stimulation techniques and talk about the underlying principles. And in the second session, we will talk more about the practical side of how a brain stimulation study could be conducted. So basically, the key or the goal of this first part of the lecture, we are going to cover a few key concepts. So first of all, if you don't have any background in neuroscience research, it is important to know a little bit about how our brain works. And so because it's closely linked to brain stimulation. And then we are going to talk about, well, in general, what is brain stimulation and how many different techniques are available as a research tool for us to use, including, I'm going to pinpoint 3 different techniques. One is called transcranial magnetic stimulation. The second one is called transcranial electric stimulation. And then a further development, more recently, is the transcranial focused ultrasound stimulation. So after covering all these techniques, I'm going to talk a little bit more about how could we compare and contrast these different stimulation techniques and how we could effectively identify the most relevant tool for us to use in research. And then at the end, I will cover a few ethical issues as well as the pros and cons of different techniques. So this is a very casual presentation, so if you have any questions, please stop me and then just ask questions for clarification. And make sure that you feel comfortable because, I mean, if you don't have the background, then maybe there are quite some new concepts that I will bombard you with the different things. So just let me know and then we could adjust the pace because we are in a relatively small group. So first of all, if you haven't learned anything about the human brain before, so I'm going to just recap a few key concepts. First of all, the human brain, every one of us should have one. It normally weighs about 1.4 kilo, like 1,400 kilograms, 1,400 grams. Relatively, male have a slightly heavier brain, and the female have slightly lighter brain. But it's not due to any gender differences. It's mainly because of the body size. So the brain is relative to, like the weight of the brain is usually relative to the body size. So it takes about like 2% of our own body mass. However, if you count or calculate the amount of energy it consumes, it receives more than 20% of the total body oxygen consumption. And if you count about like the percentage of body glucose use, it takes about 25%. So relative to its weight in terms of our body mass, it consumes a huge amount of energy in order for us to function properly. So just some indirect index to show that the brain is a very important organ because it controls essentially every kinds of functioning we are doing right now. Breathing, sitting, seeing things, listening, and also understanding language, comprehending, memorizing, you name it, it is done by the brain, usually. However, despite its complex functions, the basic unit of the brain, we call it the neuron, is highly homogeneous. So this is a typical neuron. How does it look like? So this is called the soma, or, well, soma is in Latin. This is a cell body. You see lots of tree-like, you could call it legs, which is called dendrites. They are the terminals that receives information from the surrounding environment. So there are, well, usually the neurons doesn't exist alone. So there are groups of neurons, millions of neurons that constitute our brain. So they are highly organized and they are interconnected with each other. So they receive, a typical neuron could receive more than 1,000 inputs from the surrounding neurons. So these dendrites, the tree-like legs, are the input. And then there is a very simplistic but sophisticated neurochemical processes that determines whether a neuron, a brain cell, will fire or not. If it decided to fire, then the information will pass through this long branch called the axon to deliver an impulse to what we call the terminal bulb. So this is the output of the neuron. So it gathers information from surrounding, do some computation, and then if the inputting signals are strong enough, then this neuron could decide to fire, and the firing will pass through this axon and then pass on to some other neurons, or maybe through very long-range connections to our muscles, to different receivers, receiving organs, to control all sorts of different activities, including movement. So you could control your muscle, you could control your secretion of internal hormones, or some other things. So again, there's a wide range of functions that could be done by these kind of connections. So totally, there are, the shapes of neurons varies from different types. But in general, you could divide a neuron into the dendrites, the cell body, and the axon. So basically, they are more or less the same. You could classify all the neurons into roughly like 12 different types of neurons. They perform only one function, which is to gather information and then to decide whether to fire or not. Okay? So this is a very, very simplistic model or simplistic scenery where you could reduce brain function into one thing called action potential, which is the firing of this neuron. So you could view each neuron as a computational unit. So because it receives input, it submits all the information, and then when it reaches a certain threshold, depending on a certain rule, then it fires. All right? So I will demonstrate this a little bit later. So the firing of a neuron is called an action potential. It follows a typical shape like this. So the action potential only exists here at the exit of the cell body, at the entrance of the neuron, of the axon. So once it leaves the axon, it remains the same. So it propagates along the axon via some neurochemical mechanisms that I'm going to touch a bit later on. And so you could view it as something like this. It is an electrical impulse that travels through the axon and reach the terminal. Okay? And if you use or put two electrodes to measure the change of voltage, it's always like this. This is a very typical action potential, or the shape of the curve. Why is this so important? Because for you to think about how you could stimulate the brain, you need to, in fact, try to trigger an action potential in the neurons. So that's the basic understanding you need to have. So how does it work? So how did the cell decide to fire or not? So basically, there is something called membrane potential, which is meaning that at rest, inside the brain, inside the neuron, it remains negatively charged. So if you count the number of positive versus negative charge, inside the cell body is always negatively charged. And so I said that the dendrites receive inputs from the surrounding neurons or from some sensory organ. These signals exist in terms of electrical signals. So again, it is some kind of charge that enters into the cell body. So imagine that the cell itself, the neuron itself is negatively charged. If there is some positive charge that enters into the cell body, that it will reduce the charge. Here, the axis is from -70 to +40. So imagine that there is some positive charge that enters into the neuron, that it will increase the potential, or what we call the membrane potential within the neuron. So when it reaches a certain threshold, this value is normally or typically at -55 microvolts. Then it will trigger a chain reaction that then there is no return. So there will be definitely a firing. So this is the threshold, and then so you could imagine when all the positive charge rush into a neuron, those are the information. And then once the neuron accumulates enough positive charge, then it will decide to fire. So this is the basic mechanism in which a neuron works. So far, so good? Okay. So how does it work? This is the complex picture that you don't necessarily need to understand all, but I just want to give you an overview. So basically, at rest, there are some mechanisms to maintain the negative charge within the neuron. Okay? So there are some structures that exist at the membrane of the neuron that exchange some ions in and out of the neuron. So the positive neurons or the positive ions will be transported out, and then the negative ions will be transported in. So that's the way. It's like a pump. So to keep the positive charge out from the neuron. And so this is the resting state. And then there are something called ion channels. So those are structures that could be opened depending on the voltage of the membrane. So this is a cell membrane, and then these are some channels that could allow the ions to rush in or sometimes go out. But here, all we are talking about a channel that prevents the positive charge from entering into the neuron. Okay? So I said that when the dendrites receive information, some positive charge will be enter into the neuron. So when this happens, this is called depolarization. So at rest, the neuron is polarized so that it remains at -70 microvolts. But once there is some positive charge that enters into the neuron, then it will increase the potential, all right? And this could be considered as a stimulus. So these stimulus that change the membrane potential will essentially change the structure of the ion channels and allow some more sodium ions to go in, all right? So that further increase the positive charge inside the neuron. So this curve further goes up, all right? So once it reaches a certain threshold, then these ion channels, these sodium channels will just all in a sudden, they all opens, allowing all these sodium ions to rush in, all right? So that will increase, that will result in a huge surge of potential or increase in positive charge inside the neuron. So this results in the increase of electric signal at the first point of the axon, all right? And at a certain point, then these channels will be closed. And then there's another ion channel called potassium channel that opens because potassium ions are being kept inside the neuron all the time. But then now, when the potassium ion channels just rush out, it will lead to a reduction of charge because the positive charge leaves again. So it is all the summation between the positive and the negative charges, or positive and negative ions. Because now the positive charge leaves the axon, leaves the neuron. So the potential reduces again. So there is some overshoot, and then it will return to the baseline. So basically, this is how an action potential is being resulted in the axon. All right. So it is the interplay between the charged ions that flows in and out of the neuron that result in this signal. Okay. So just a quick summary. An action potential is the chain reaction of electrochemical changes along the axon. So this is like a domino-like reaction. So at the first point that there is one action potential, it will lead to the same changes at the surrounding regions of the axon. So it propagates through the axon, all right? And these movement of electric charges are regulated by these voltage-dependent ion channels, okay? The other important thing is that it follows an all or nothing principle. So which means that once the threshold is reached, then definitely an action potential will be resulted. And the neural signals, in fact, are being encoded by the frequency of action potential. So I'm going to demonstrate this to you. So this, imagine there's one cell that is at the primary visual cortex. So it receives visual input. The function of this cell is very simple. It only detects whether a vertical line exists or not. So because when you view your image through the eye, then through the optic nerve, then there are some information being passed to the primary visual cortex. So how could it encode the orientation of a certain stimulus, a visual stimuli? So you need to know that first of all, there are always some kind of background action potential. So even if the cell does nothing, occasionally it will fire. So it's not like, okay, if there is no stimulus, then there is no action potential. The brain doesn't work like that. So even if you are thinking of nothing, you are idle. In fact, there are all sorts of activities going on in your brain. So even at the single cell level, it's the same, all right? So when you see a stimulus, if this is the target that this cell is really sensitive to, then it would lead to a chain of action potentials, so a very frequent firing. But then even for stimuli that are close in terms of the physical property, it will still fire somehow. So it's not like all or nothing. It doesn't like, okay, I only fire when I saw a 90-degree vertical line. It doesn't work like that. So it follows a certain kind of distribution that determines the cell's, what we call the response tuning curve. So by looking at this, this is the frequency of firing for different orientation of the line. So when it comes to a horizontal line, then there is a very, very low rate of firing. But when it changes to more and more proximal to the target orientation, then the firing rate just goes up. So by simply looking at the frequency of these action potentials, the cell could represent somehow what kind of stimuli is looking at. So you could generalize this to some other form of sensory perception. So in auditory cortex, there are some cells that is sensitive to the vibration of a certain frequency range. So when you're hearing some very high-pitched noise versus very low-pitched sound, then there are some differentiation between the neuronal population that is responsible to analyze this kind of frequency of auditory signal. So the same goes for visual stimulus. So this is the most basic form of orientation processing. So there are other cells that is sensitive to edges, to corners, to curves, and by stacking layers of neurons, you could kind of abstract the information from the most basic visual property to things like objects, to faces, to the identity of faces. So it's like a hierarchical organization. Okay, so this is a very rough picture on how the brain works, all right? So just again, I want to further elaborate on the neural signals, how the neural signals are encoded by the frequency of action potential. So I only show you what happens when we have one single cell. But you know that we have, I didn't find the number, but the huge amount of neurons that exist in the brain. And their activities are highly coordinated. So imagine that this axis, one line represents the activities of one cell. So now we are talking about a group of different cells. And the dot represents the action potentials. So somehow even if the subject or if the animal that we are making these recordings are doing nothing, the brain cells do fire in patterns. So if you aggregate all these activities along the y-axis, you will see something like this. So these are the periodic oscillations of brain activities. And we know that these oscillations are meaningful. So somehow, depending on the frequency of these curves, it could represent different mental states, for example. Like if you are doing EEG to look at the sleep, so there are different stages of sleep that you could look at this slow wave, what we call, or slow oscillations of the brain potentials. Or when somebody is performing some cognitive task, memorizing or doing some working memory, then at a specific frequency range, then you will see these oscillations, all right? So this is another way, yep.
Can I ask the y-axis of the graph? Is this spatial or?
This is spatial. So one unit represents one cell, the data from one cell, all right? So the key message is that there are neural oscillations that is also meaningful if you count not only one cell, but a group of cells, what we call neuronal mass, all right? So if you do things like EEG studies or MEG studies, you could, somehow analyze your data with this kind of time frequency plot. So this, I'm just using it as an example. So it is not important what kind of task is being performed by the subjects, but here this line represents the stimulus onset. So this is the time point when the subject is being asked to perform some task. So actually this is a working memory task. And then if you look at the one recording site and then look at the frequency of your brain signals that you have, so you divide into different frequency, and then you plot the amplitude or the power of this certain frequency, you could have this kind of plot that represents the kind of responsible frequency that is being important in performing the task. Okay, because here it represents a reduction of power, so there's some change of potentials in this 10 Hertz range. And then you could do some kind of comparison, for example, healthy control versus schizophrenic patients, and this is the difference between the two. And then you could make some inference on, for example, on this frequency band. So at around like 500 milliseconds time, at a 10 Hertz range, there is some significant differences between the schizophrenic patients and healthy subjects. So this is kind of typical EEG results you could see. The key message is that we could look at these neural oscillations, because when we talk about some kind of frequency, we are assuming that the signal will oscillate like a sine wave or cosine wave. All right? So having talked about all these kind of very basic brain functioning, so please bear in mind, because we will get in touch with all these concepts later on. So what is brain stimulation? A very, very simplistic definition is that this is, by whatever means you try to perturb neuronal activities. So if you look at the history, in fact, you could trace it back to the early 40s, I would say, or 30s, that already there are some researchers trying to do this kind of research. So I have a very short video that we could take a look at. So Dr. Walter Penfield, in fact, is a very, very famous neurologist who founded the Montreal Neurological Institute. He is the kind of pioneer.
During my life as a brain surgeon,
It's a little bit
necessary to operate on a good many men and women, a good many hundreds, and to expose the brain under local anesthesia with the patient conscious. In those operations, it is a useful, practical procedure to stimulate the cortex electrically.
okay. So I think because of the quality of the video is a little bit difficult to hear. So he just said that when treating epilepsy patients, so the way they treat it is by surgically remove a certain part of the brain, which is the center of epilepsy. And he said that it is important and also useful during the operation to stimulate the brain, because the brain is so important. You don't want to cut away a certain part that carries important function. For example, if the epileptic center is located near to your language center, so that by removing this part, that will influence your daily function very much. Or if it is close to the motor center. So it is important to kind of like map the function of the brain tissue that is being removed or that is going to be removed. So what here the video is showing is that how this kind of operation could be done. So usually this is done only by local anesthesia, so which means that the patient is awake, because you need them to be awake in order to perform some tasks, right?
These are not experiments. In that process, we stumble quite accidentally on the fact that there is recorded in the nerve cells of the human brain a complete record of the stream of consciousness. All those things of which a man was aware in any moment of time are recorded there, and all the sights and sounds which he ignored and the thoughts which he ignored are absent from that record. The operations are carried out under local anesthesia, so that the patient is conscious and can talk.
So the patient is here.
He does not feel anything when the electrode touches the brain.
So this is the brain. So they open the skull
It is as though
and then use some electric probe electrodes to probe
or a strip of film,
the brain.
and he relives a period of time. When I talked to them afterwards about how it seemed to them, they say it is much more real than
So,
any
oops.
memory. It is as though they were re-experiencing it. I believe you're about to hear the actual words of patients in response to this stimulus, which they were quite unaware of while they were in the operating room. You will hear them as I heard them.
I have a sudden feeling as though I lived through all this before.
Now I see them. They're laughing, my friends. And I'm with them in a house in South Africa. They're my old friends.
I hear children's voices. I hear them down along the river. I'm sitting in a railroad station in a small town in Kentucky. It's winter and the wind is blowing outside, and I'm waiting for a train.
Okay, so could you hear what they're saying? These are the recollections of the patients undergoing this surgery, and when the doctor tries to stimulate a certain part of the brain, what they experience. So sometimes they said that I hear some voices, like I hear some children's voices, one of the patients said, and then some people say they will see something, or they will, it will trigger a certain kind of memory or some smell. So basically, he is the pioneer to demonstrate that if you use an electrode to kind of like stimulate the cortex, you could revive all sorts of experience in alive human subjects. Okay. So, why is this so important that we want to do brain stimulation? Because now we have all sorts of techniques, like you could use EEG, you could use MEG, you could use fMRI to see how the brain works. But all these techniques are only correlational. So this is not like the topic of this workshop, but the key here is that if you do brain stimulation, you could make very strong causal claims on the kind of functioning you want to examine, because you are actively trying to induce an experience or a phenomenon in your subject's brain. So the only key factor that you've put in as an experimenter is brain stimulation, for example. So unlike all other neuroimaging techniques, you could make causal claim. Okay? And again, how does it work? I need to take back this action potential graph again. Because you remember there's a threshold here. So in one way, when you stimulate the brain, you could either reduce the threshold or increase the threshold, so that the neurons will be either less likely or more easily to fire. So that's the excitability of the neurons. So this is only one way. The other way is that you could, by some means, try to inject some positive charge into the neuron to trigger an action potential. So basically, all the brain stimulation technique should, in theory, work either by one of these principles. All right? Okay. So from now on, I'm going to talk about the more specific techniques. So before that, do you have any questions? If not, then we go on. Okay. So the first technique I would like to introduce is called transcranial magnetic stimulation. So you need to know some physics in order to understand how it works. So it's basically based on physics principle. You need to know what is eddy current. All right. So if you have some, I don't know, I think third level physics or DSE physics, or you will be able to understand. So if you have a coil here, so just like every single motor, when you pass alternating current, then you could generate some kind of electromagnetic field by this coil. And then, why a alternating current is important? Because then the magnetic field will change the direction. Because when it is like when the current is like at the positive side of the curve, then there's one direction of the electromagnetic field, and then it reverses. So the changes of this electromagnetic field on a conductor will induce something called eddy current. All right. So we could indirectly use a magnetic field to induce current at a certain site. All right. So basically, TMS, in short, it is work like this. You have a coil, usually is in the shape of 8, because then you could really focus all the magnetic field into a very narrow area, right? This magnetic field usually is not that much absorbed by the scalp, because there is no or not much electromagnetic substance there, because it is only like bone, mainly bone. But then, so that's why this magnetic field could pass through the scalp to reach the cerebral cortex, okay? And the cerebral cortex itself is conductive. Remember that when we talk about action potential, when we talk about the neuron, it is all like in an environment that the charges could be moved freely, right? So this change of magnetic field could induce an eddy current at a certain site at the cortical level. And this could trigger an action potential. But the key is that the generated magnetic field is very, very strong. So it's about 1 Tesla. So Tesla is the unit of magnetic field. I think it's like, I don't know, the background magnetic field of the Earth is like 0.00001, something like that. I haven't checked the exact figure. So it's like 1,000,000 times of the Earth's magnetic field. So it's very, very strong. So that's also one key or one precaution if you go to a TMS lab. And then if you are going to do some experiment, remember, remove all your credit card before you do your experiment. Otherwise, once you put the coil near your wallet and then you have 1 pulse, everything's gone. So that's like one sidetrack thing. So it usually, a coil generates like a magnetic field of 1 to 1.5 or even higher Tesla. So then this magnetic field in turn induce an eddy current at the cortical site. All right. So this eddy current is an injection of positive or negative charge that triggers neuronal firing. All right. So this is a typical coil, but then I have another video to show you. So this is a typical way or
Welcome to our laboratory. Today we're going to show you how the transcranial magnetic stimulator works. So first thing, Yoshi's going to put the magnetic coil over top of the area of my motor cortex that's responsible for the muscles of my hand and forearm. Once she's there, she's going to deliver a magnetic pulse. Oh,
so you see the hand?
my arm twitch there. And we can also see this response over here on the oscilloscope because we've measured it using the EMG electrodes over the muscles of my hand and forearm. Now what we're going to do is move that magnetic coil over the span of my motor cortex from one side all the way over to the next. And what you'll see is muscles slowly recruited from my hand up to my elbow, shoulder, right leg. And after she switches hemispheres, we'll see some of the left leg come in and then down the muscles of my left arm. So here we go. So right hand,
so hand, and then
a little bit of elbow, some
now
shoulder and leg,
you'll
still
see
right
that
leg,
now it
little
comes to
response
here,
to my left leg there, we've switched hemispheres,
and then now
more leg, on the left side, left shoulder, left elbow, and left hand. Thanks for joining us.
oops. So you see, this is like a very good demonstration on how TMS could work. All right. Let's see if we could do something similar today. I don't know. So, so these are called single pulse TMS. So basically, when you press the button, so there is one magnetic pulse that passes through the skull to reach the cerebral cortex, and then that triggers one response. Okay? So it triggers the neuronal firing at the spot of stimulation. So this is what the girl has just done. So you applied a TMS pulse at the primary motor cortex, depending on the region, and it could send a trigger through the cerebrospinal thread to the effector muscles, to the arm or to the hand, and then you could then measure this change of muscle tension. So this is also called TMS MEP, or motor evoked potential. This is one way already to do research with TMS. Because if you think about, there are some neurodegenerative disease or even simply aging that could result in the change of this evoked potential when comparing different groups. Okay? So this is a way to probe how the brain responds to triggers that determines what we call the motor threshold. All right? You could also put the coil to other places in the head. For example, you could put it at the occipital cortex, which refers to, or when you generate the pulse, then the pulse will reach the primary visual area, primary visual cortex. And then you could induce some phosphines. So phosphines are perceived stimulus without the actual existence of a physical stimuli. So that means your eyes, your retina didn't receive any light, but then you think that you saw some light. So you could also induce phosphines by closing your eye and then just press on your eyeball. Everyone does that when you're small, right? So these are called phosphines. And you could use this magnetic pulse to induce this kind of stimulation. You could already, again, use this property to do some research in terms of the visual system. For example, if you could try to change the color of the phosphines by adaptation, or you could try to use a certain frequency of stimulation to induce different types of phosphines. It also appears in different, what we call the visual field. So depending on the site of lesion, you could, not lesion, but the site of stimulation, you could check if you saw these phosphines at different locations of your visual perceptual areas or visual perceptual space. So that's also like one way to map the retinotopic organization of your visual areas. So these are more basic experiments that you could think of. You could also do more sophisticated stuff. You don't necessarily need to limit yourselves in terms of the motor function or the basic primary sensory functions. You could also do what we call now online TMS on perceptual or cognitive processes. Okay, so for example, if you want to study attention or working memory or even memory, you could have a certain task, and then at a certain time point, you deliver a TMS pulse to, well, there are different forms. You could use a single pulse, or you could use a double pulse, or you could use even short burst. So different types of stimulation protocol will result in different effects. The effect is highly task, context, and time dependent. So it's really hard to say what you are going to get if you have a task. So you need to, first of all, you need to know the literature on how, because if you just, you don't have any experience and then you just started, you need to read the literature in order to see what people have done before. One key factor is this time-dependent influence, because the TMS pulse could be coincide with the presentation of the stimulus, or it could be a few milliseconds beforehand, or a few milliseconds afterwards. So by putting the TMS pulse at different time points, you could disrupt different types of processing. I know I'm talking a bit vague here, because it's really specific to the kind of function you are working on. So I could not provide a very, very concrete example. So anyway, so for single or double pulse, you could imagine that it is like injecting some noise to the system. And it is like an interruption of the processes. Because when the cell fires an action potential, there is something called a refractory period, which means that it's more difficult. Well, there's one absolute refractory period in which no more action potential could be induced. And then there's another relative refractory period, which it is more difficult to induce an action potential, which means that if you lead the neurons to fire prematurely, and then you have the task, then the ability for the neuron to process the information related to this task will be reduced. So it is kind of like a disruption of the system just right before the stimulus is presented. So that's why the timing is important, because if you do it too early, then the neurons have enough time to recover and to perform the original function. But then if it is too late, then again, the neuron has already encoded and processed the information and then to deliver the output. So the temporal precision of your TMS pulse is really like critical to whether you could receive an effect or not. All right? And then for burst, because when we talk about burst, we are talking about some regular pulses delivered at a certain frequency. So it depends on the oscillation frequency, because by doing bursts, you could somehow affect the overall oscillations at a certain frequency band. So this is again the more complex. Usually for online TMS, people are doing single or double pulse, because for bursts, it's more difficult to understand the outcome and to relate it to a kind of like cognitive mechanisms. Okay.
Explain the burst a little more. So the burst is different from the double pulse and
So the burst means that you delivered more than one pulse regularly. For example, if you have the pulse, like you have 10 pulse every second, then you are having a burst of like 10 hertz, right? So it depends on how frequent you are stimulating. Either it could be very frequent, like at 50 hertz or so. So yeah. All right. So this is called online TMS, which means that you deliver the pulse when the subjects are performing a certain task. But it could also be offline, which means that you first stimulate the brain, and then right after this stimulation, then you ask the subjects to perform the task. So then the key is that this stimulation protocol should last for a duration. I mean, the effect of this stimulation should last for a duration that covers the whole experiment, right? Otherwise, if the effect degrades at like 10 minutes after stimulation, and then your task lasts for an hour, then you probably only could observe an effect at the first 10 minutes, and then there is the effect will be diluted or even like become insignificant. So you need to know the effect, but for TMS, when you do repetitively, which means that again, before the experiments, you delivered a chain of pulses at a certain frequency range, and it is believed to reset the phase synchrony of neuronal mass. Because remember, I showed you this picture of a group of neurons that they fired together up and down. So those are called phase synchrony. So normally, when the brain is doing something, then the neurons are very noisy. But then if you put or inject some very regular pulses by doing RTMS, then you could reset this phase synchrony of neuronal mass. So by using different frequency of this RTMS, you could achieve different functions. For example, for 1 Hz RTMS is observed to be inhibitory, which means that when you put the coil over a certain region, for example, like the primary visual cortex, and then you apply 1 Hz RTMS, then you could temporarily reduce the activities of the stimulation site. Okay? And the underlying mechanism is believed to be something related to the long-term depression of the synaptic connections. And conversely, if you do 10 Hz RTMS, is observed to be excitatory. And then it is believed that, or it is now observed that the 10 Hz RTMS is facilitating long-term potentiation, which means that it enhances the connectivity between the different neurons or the synaptic strength, so that it helps the subjects to, or the target site to perform the task better. And then there's something now called a theta burst, because normally if you do either 1 Hz or 10 Hz RTMS, you need to stimulate the subject for over 30 minutes or even 40 minutes. So the protocol is rather long. But now people observe that if you use a certain specific pattern of stimulation, so if you do it like 3 very rapid pulses as a block, and then you separate the block with very short interval, for example, 200 milliseconds for block. If you stimulate using this kind of frequency or pattern to stimulate the brain for, let's say, 6 minutes, you could have an equally effective stimulation protocol as compared to the 10 Hz or 1 Hz RTMS that is being stimulated for 40 minutes. So that's why it is now becoming more and more popular. Okay, so this is about TMS. So remember, TMS could be offline using RTMS or online. And then the difference between offline and online is that for offline stimulation, you always stimulate prior to your experiment, which means that if you think about control, either you could do it like in a between subject design. One group receives stimulation, one group receives no stimulation, and then you compare the group performance on a certain task. Or you could do a repeated measure, but then the stimulation would be days or even weeks apart because you don't want to have any kind of carryover effect. And then you perform the task twice, and then you compare within subject the effect of stimulation versus no stimulation. Okay? Okay. So now, yeah.
For this slide, I wonder what is it related to the interface interval? I mean the RTI in there.
Oh, well, because it is offline, there has no relationship with your paradigm. So if you present any stimulus, that is another issue. It doesn't relate it to the stimulation itself.
So how about the RTI?
Oh, this is interblock interval. Interblock interval, which means the block consists of 3 rapid pulses, and then these blocks are separated by 200 milliseconds. Okay, so it's a da da da, da da da, and then this interval is 200 milliseconds. Okay, so now I'm going to go through another technique which is called transcranial electric stimulation. In fact, it's being done very, very long time ago. It could be traced to even Roman time, when there's a guy called Scribonius Largus. I think it should be pronounced in Latin, so I'm not very good. Anyway, this guy is very interesting because based on some previous literature already known in Greek time, people believe that, or people observe that when you put a torpedo ray, which release some electric current at the head of some patients, then you could treat some problems. For example, if people have headache, you could try to put this fish at the forehead, let it deliver some electric current, and then somehow, for some reason, the patient could be treated. Also, some chronic pain issues, gout, So this guy is the first guy in history that formally reports in the literature that he used a living torpedo fish putting on the scalp to kind of stimulate the brain and to treat some problems. And then later on, after the invention of electric batteries, so this guy called Giovanni Aldini, he again, based on this knowledge that if you pass some electric currents to the head of a patient, then you probably could treat things like headache. And he used the newly invented direct current battery to do the same thing. So basically, he is the first guy to kind of do transcranial electric stimulation, so to speak. So how does it look like? Basically, you have a machine called a constant current stimulator or constant current generator, which delivers a current to the scalp of the subject. You have two electrodes, one positive, one negative, right? So you put two electrodes at the scalp, and then you pass this current. So in theory, you are passing a current through the scalp bone, depending on the arrangement of your electrode. So anodal means that there is like positive current flowing from this to here, so that's the current direction. Cathodal means the reverse, like negative charge flows from here to here. So here is the active electrode. So you could either do what we call the anodal stimulation, which means you are injecting the positive charge to the underlying brain region, or you could do something called a cathodal stimulation, which means you increase the negative charge. Remember again, so here we have the threshold. So when you do this transcranial electric stimulation, you are in fact trying to affect the threshold of the action potential. So if you are injecting positive charge, when you're doing anodal stimulation, you are reducing the threshold because there are more positive charge. So it's easier for the cell to fire. But then for cathodal stimulation, you are increasing the threshold, which is becoming more difficult for the cell to have an action potential. So anodal, cathodal is all relative. And then, all right. So there are also 3 different subtypes of transcranial electric stimulation. So first of all, you could do transcranial direct current stimulation, which means that the direction of current flow is just one way. There is no alternation. So you are constantly supplying some charge, positive charge to the anode and then negative charge to a cathode, so that the respective brain site will be influenced. And there's another thing called transcranial alternating current stimulation, or TACS, which means that you put 2 electrodes and then you are having some alternating current, applying alternating current between the 2 stimulus sites. And then there's a third class of TES called transcranial random noise stimulation, which is even more, you could say, confusing, because you are simply putting some noise into a system. So you are putting some noisy current, like if you have a low noise current or mid noise current or high noise current, this is the noise amplitude. So these are some current that you put into the respective brain area. So for the transcranial alternating current stimulation, you are trying to affect the phase synchrony of the oscillations of the 2 different sites, because you're applying the current of the 2 sites to be out of phase or in phase, depending on the shape of stimulation. So by doing so, you could, for example, you could test this phase difference and its influence on the accuracy of a certain task. By plotting this phase difference and the accuracy profile, you could find that optimal performance is being observed somewhere here. So when there is a, like, let me see, 1/6 of the cycle, and I don't exactly remember, is it like 90 degrees? So when the 2 alternating currents are completely out of phase, so which is like half a pi in terms of the radian, then the performance is worse, which means that these 2 brain regions are kind of like coupled together if there is no stimulation. So this stimulation is disrupting the crosstalk between these 2 regions, and that's why it's reducing the accuracy of the performance. And here, if you could also look at the amount of noise being injected into the system, and how well the subject could discriminate some visual stimuli or degraded visual stimuli. And by looking at that, you could see that, okay, if you somehow inject a medium level of noise into the primary visual cortex, V1, you could improve performance. So this is somehow linked to the understanding of how the brain works. For example, there's some sort of a stochastic model of visual perception, which also takes into the account that noise are meaningful. So by increasing an optimal level of noise into the system, then the brain could perform better. So that's the current understanding. So basically, if you want to do TRNS, you need to have a good model on the task that you want to test in related to the amount of noise, because you could not just say, okay, I just stimulate and then by doing some tasks, inject some noise, whether it improves or not, it doesn't tell you anything, okay? So these are more, you could say, more advanced form of research protocol that you could use. Okay, so this is TES in general, so the 3 different big classes. So in the afternoon session, we will talk about more the technical details on how you could design an experiment. And then, okay, the last stimulation technique, which is more recent, is called transcranial focused ultrasound stimulation. So now you imagine that if you accompany your wife, or if you go for a pregnancy test, you do the ultrasound, that thing, that transducer, in fact, could also be used to stimulate the brain. It's not only for imaging, but the power is different. So first of all, I mean, in fact, we don't need to go through all the technical details. It is just showing that if you put a transducer, ultrasound transducer at the skull, and then you measure whether the ultrasound pulse could still reach the brain tissue, the profile, like when you are doing it in free space versus doing it in like above the skull is very similar, although there is a reduction of power. But you could still do focused stimulation. And then here is showing that the effect is very, very focused, very, very narrow. So here is the brain region about the site of stimulation. So there is so far only like one study that is being published that use this technique. So just for your knowledge, that you could keep in mind that maybe, I don't know, in 5 years' time, it will be another popular technique that could be used because ultrasound is, again, very safe, and then probably there's not so much side effect as compared to TMS or TES. So again, after covering these techniques, you could see the comparison of different stimulation methods in terms of the temporal resolution, which means the effect lasts for how long. So it is in a log scale, so you could be as quick as, like the effect could be as quick as 1 millisecond or some seconds or some minutes or even hours, depending on the type of stimulation protocol that you use. And then the other main concern is that the spatial resolution you want to achieve. So for TES, you will influence a relatively large area of the cortex. But then the TMS is more focused. TFUS is even more. So that's why the potential for this ultrasound stimulation is quite nice. And these are all called NIBS, so non-invasive brain stimulation, which means that so far, at least for our current understanding, there is no long-term side effect or no long-term bad effect on the brain, on brain function. So it's safe to be used on human subjects. Okay. Okay, so applications of brain stimulation. Already, I mean, you could imagine there are lots of different studies in the cognitive neuroscience field that taps on different kind of brain functioning. So long-term enhancement of brain function in learning, some mathematical skills, for example, in looking at dyslexia, so different populations, you name it, you could almost find it. So any different kind of things. So this is interesting in terms of our understanding of the human brain function.
Excuse me, I have a question. As far as to your understanding and your knowledge, have you already seen any studies about the imagination?
Imagination.
Yeah, because it is not some cognitive task, like attention, memory, but anything about imagination.
I haven't, to be honest, I haven't followed the literature on imagination, but I guess you could do a quick Google search and then I would imagine that someone must have done something in terms of, for example, creativity. Yeah, that could be, or maybe I'm wrong, I don't know. That's something for you to explore, I would say. Because I'm not working on this area, so, but yeah, I think you could easily, if you already have some kind of experimental protocol, you could easily convert it or modify it to become a brain stimulation study. So why not? Yeah. So that's on the research side. And then on the application side, or more clinical application side, there is already FDA-approved treatment in terms of depression. I think there are also companies trying to obtain FDA approval for Alzheimer's disease in combination with some cognitive training. But I haven't heard anything like formal results yet. So, I mean, if you search, then there are some commercial companies providing this kind of treatment. And even in Hong Kong, there are already people doing this kind of TMS treatment for Alzheimer's. So it's getting more and more popular. And the last thing I want to talk about is that if you look at, well, the TMS is more sophisticated. You probably couldn't build a coil yourself because, first of all, your electric bill will be extremely expensive, so probably no one will do it at home. But then if you look at the TES, the transcranial electric stimulation, it's quite easy to do. So if you go to YouTube, just search TDCS DIY. There are already like 2,300 something, almost 2,400 results, which means that people are, in fact, if you go to Reddit, so if you know that, the website for kids, for nerds, there are a long thread discussing how you could build your own TDCS machine, because the concept is really simple. You could use even a 9-volt battery to build a machine that delivers the required current for stimulation. But again, so this is a new trend called neurodoping. So you are kind of like taking some excitement drugs for your brain, or in combination with different things, people also term it called brain hacking. So try to hack in your brain to improve the function. Because it's so easy and so convenient, lots of people are doing it. But then there comes for ethical issues, because again, although you are only responsible for, if you do it yourself, and then you are responsible for what you've done to your brain, but again, it is ethical or not, is a debatable issue. Brain stimulation itself is not suitable for everyone. So there are contraindications. If we do a research, we need to first have at least some sort of screening for the subject before we could decide whether one person could be recruited as a subject or not. First of all, there are potential side effects. If you don't do it properly, you could potentially induce a seizure in the subject, which means that for subjects who have or who has a history of epileptic seizures, you should not do any kind of brain stimulation with them. Also, for people who have psychiatric history or neurological disease, you should not stimulate them, because you don't know what will happen. If a subject went into a stroke, who's responsible? Although there may be some kind of like before the experiment, there already some vulnerability, or maybe there's an aneurysm, but then who knows? And it's difficult to say who is going to be blamed. And all these potential side effects, for example, if you do TMS for a long time, then maybe after all you will have some kind of headache. If you do TDCS, if the current strength is too high and then the duration of stimulation is too long, you could burn your own skin. And there is also very largely unexplored effects on altered mood and cognition. So the dosage is critical, and I would say never try to do it yourself. Although it's so convenient, we don't build a TDCS machine and then we go to test some subject. That's not what we should do. Okay, so I guess this is the introduction, and then any questions and comments or discussions?
Can you talk about the TMS? There is a discrepancy in the function between one Hz and 10 Hz. One of them is depression, and another is potentiation.
Mm-hmm.
So do we have is there any explanations why the different intervals will trigger so different results?
Yeah, there are some evidence suggesting that the different hertz of stimulation will trigger different release of neurotransmitter. And 1 Hz is related to GABA, I think, which is inhibitory in nature. And then the 10 Hz is triggering something related to the acetylcholine, I think. So different types of neurotransmitter are being somehow indirectly released by different frequency of stimulation. But I think the actual mechanism why the different frequency will trigger this different neurotransmission is still unknown, I would say.
Trial and error.
Yeah.
And about the last technique, this ultrasonic one, you mentioned it's new, but do you have some idea why it works? Because it would go beyond what you talk about as action potentials of those kind of things.
The mechanism is very funny. Is believed or is hypothesized to move the neural tissue itself. So the ultrasound will create very tiny displacement of the neural mass, which is like you try to squeeze your brain, and then it will trigger some reactions. So I think right now a team in Stanford is really trying hard to use animal models to pinpoint why it work or not. Yeah.
Another question is the comparison of the TMS and the TES. So we know TES is cheaper and easy to actually operate. However, from the scientific view, the reliability and so for the cognitive experiment, do you have any suggestion which one should be used? Because we have both.
Yeah. The factors you've pointed out is one thing. So, like how easy you could set up an experiment to stimulate the brain is one thing, and then potential side effect is another thing, because TMS is more accurate in terms of the spatial location, but there are also relatively more aversive effects. So people generally don't really like it, because if you stimulate the prefrontal cortex, then you will also stimulate the ocular muscles, the muscles around this region. So it could be if the stimulation is near the vagal nerve, you will also have some sensations. So in terms of the pain sensations you might have, the chance that when you are doing a TMS is much higher to have this kind of pain or uncomfortable sensation as compared to TTCS. TTCS usually, I've tried once myself. Just at the very beginning, you will feel a little bit of tingling sensations, but during the stimulation, you won't feel anything. So it's relatively easier for people to accept it, to perform it, or you could say that to come back again if you want to do multiple sessions of experiments with them. But then the downside of TES is that the area of stimulation is not that precise. Also, if the target location is not at the gyro site, so it's in the sulcal site, it's more difficult to stimulate. Although there are more advanced techniques that, you could try to overcome these issues, but it takes some time for you to master the technique in order to get good results. So I would say that for both TMS and TTCS, they are not as easy as you just design an experiment and then you run it and then you see whether you have effect or not. There are many, many subtle parameters that you need to fine-tune in order to make it work. And I would say I'm not the best person to provide you with this subtle parameters, because I'm not really an expert in doing this kind of research. So hopefully in the later workshop, then someone will come and cover those things. So I'm going to give you like an overview, a general picture, some theoretical background, and maybe some hands-on experience. Yeah? Some more questions?
There's no more questions. Let's wrap up our sections, and thank you, Kenneth.
Thank you.